Hello, I'm going to go through the second year 10 lesson for those of you who couldn't come to school. So, what we did was we looked at abiotic factors, non-living factors, and how it may affect living things. So there's a few blanks here, so I'm just going to go through this. So yeah, light intensity, that's an abiotic factor, and plants and algae need light for photosynthesis. Whenever you see light in a question, think photosynthesis. Next one it says, if too high, it causes enzymes to denature. Right, well, pH can cause that. Look, pH is further down, so yeah, it's temperature. High temperature causes enzymes to denature. Low temperature causes enzymes to not function because they collide without enough energy. Okay, it's like that clap is not a good clap. You can't see me. I haven't got my camera on. Anyway, if you could see me, I did a slow clap and there wasn't enough energy to make a clap noise. Moisture levels, yeah, so when things are dry, that can desiccate living things. Desiccate means to dry out. It's not a word that's in the syllabus, is desiccate, but it's a useful word to know. pH, again, enzymes work best at an optimum pH. Be it too acidic or too alkaline, they will stop working. Remember, protease in the stomach, its optimum pH is about pH 2 or 3. Okay, so anything lower than pH 2, denature. Anything higher than pH 3 or so, it starts to denature. Needed for producers to make amino acids and proteins, well, that's nitrates or nitrate in the soil. There's two minerals we need to know, nitrates and magnesium. Magnesium is needed to make chlorophyll, nitrates to make amino acids, and then we join amino acids to form proteins. Wind strength, again, it's an abiotic factor, and that can cause desiccation. CO2 levels, plants, and we'll look to the top one, algae, CO2 for photosynthesis. If a question's talking about CO2, the answer is going to be related to photosynthesis. And then aquatic oxygen levels, active animals require oxygen for respiration. But if you want to get another mark, talk about aerobic respiration. Okay, so we're going to look at abiotic and biotic factors. Abiotic factors are really easy to measure. Okay, so here we have three ways of measuring oxygen. So it says we can, in inverted commas, measure oxygen with these. And then it says which one of these can provide quantitative data on oxygen concentration. We have an oxygen meter here, an oxygen probe. Any abiotic factor that you want, there's a probe and meter that you can buy. Okay, and this costs money. You chuck it into the water and you read off the oxygen concentration. However, I can go to a pond. If I see these things, this is gammarus, a freshwater shrimp. These zip around in the water, and if you move quickly, you're going to need to do aerobic respiration. Therefore, it's an indicator that there's oxygen present. If these were, not moving or dead or not present, that indicates that there's low oxygen. Whereas these are leeches. Again, these are slow moving, and these are typical of anaerobic water, and therefore they tell me, oh, there's probably not a lot of oxygen there. But if I wanted a quantitative measure, then I need to use this. Yeah, I need to use a meter with a probe. Okay, because these just give me qualitative data. They just tell me yes or no, or a lot and less. They give me word measures. They don't give me, I can't look at these and say, oh yeah, the oxygen concentration is 38.5. Yeah, whereas this thing can. Make sense? Good. So yeah, abiotic factors, really easy to measure because you just stick something in. Then, right, so we're going to look at how we measure, how we write an answer. So it says, how would you show that the dissolved oxygen concentration changes in a pond as the water temperature changes on a sunny day? We get lots of these planner method type questions that are worth 6 marks. And this person's got 1 out of 6. They put place the oxygen probe in the water and measure the oxygen throughout the day. Also measure temperature as well. A really low level answer. 1 out of 6. This person's got 2 out of 6. They've done a proper list, numbered list, which is better. But they've put place the oxygen sensor in the pond. But where in the pond? Place oxygen sensor next, a thermometer next to the oxygen sensor. That's good. So I know where I'm putting them both. They're going next to each other. Record the oxygen concentration and temperature of the water at the same time. That's good. I'm saying when I'm doing it. I'm doing it at the same time. Repeat this until you have 3 pieces of data. I don't know what I'm repeating there. It's not answering the question how it changes during the day. So really, we are going to go through how to turn a 2 marker into a 6 marker. And it's dead easy. I just make it specific. So this is a 6 out of 6. So I place the oxygen sensor in the pond at a depth of 20 centimeters. Anything, if I place the oxygen sensor at the edge of the pond or center of the pond, I've said something specific that puts me to a high level answer. Place the thermometer next to it and leave for 20 seconds. So I'm going to put a time there. I'm obviously keen on 20 here. And I can put any number here. It doesn't have to be a correct one. It just needs to be something that makes it specific. I've controlled time. Yeah. Obviously, if I put a thermometer in something for a second, it might not have time for it to warm up. So I do need to leave it for a set amount of time. Record the oxygen concentration, temperature of the water, that's good. Take 3 recordings every 20 seconds and calculate a mean. That's good. I've kept my 20 going as well. And I'm going to discard any anomalies. 4 is really good. Yeah, that's what we do in science, isn't it? Is just to make sure I'm accurate, I take numerous readings to make sure I've not got an anomaly. Maybe I did put the thermometer in took it out a bit too quickly. It hadn't warmed up or cooled down enough, and therefore I got an anomaly. 5, and this is why we like a numbered list, because I can just say repeat steps 3 to 4 every hour for 10 hours. That means I'm getting data throughout the day. And then it says, how would you show that dissolved oxygen concentration changes? So I'm just going to show it by drawing a stratigraph of water temperature and oxygen concentration, and then draw a line to show the correlation. Okay, again, a really spot on, high quality 6 out of 6 answer. Okay, so and that's what you're going to have a go at in a minute, hopefully. So here's a tree, and it's covered partially with algae. So the green side is algae, little microscopic cells there, and the other side has nothing on it, or it's got a few patches but not a lot of algae. So there's definitely a difference here. So there must be a reason, because all the trees show this pattern. And science works by, we make an observation first of all. So I spot something. Algae seems to grow much more on the north or shaded side of a tree trunk. In the northern hemisphere, it's true, assuming it's not shaded by other trees. Okay, and then I make a hypothesis. I think the shaded side is more humid, as it is heated less by the sun. That makes sense, because the sun will be on the south side, as in the south side in the northern hemisphere gets the sun all day, or gets sunlight all day. The algae is more likely to stay hydrated on the shaded side. So I know algae want to do photosynthesis, but they're not found on the sunnier side of a tree. They're found on the shaded side of a tree. So it can't be they don't like light, because they like light for photosynthesis, but with light comes heat, and therefore maybe it just makes it drier on that side than the other side. Okay, yeah, the north side is shaded in the northern hemisphere, opposite way around in the southern hemisphere. So then I make a prediction, because there's lots of things here. So I hone in on 2 things. More algae, the biotic, I'm going to measure algae, will be found on tree trunks where there is less sunlight. Okay, so I'm going to check sunlight levels and see if that's true. Do I get more algae in places where there's less sunlight? So these are the things that I need to think about. So I'm going to go to the next slide, and this yellow pane just appears again. So here it is. So things we need to decide. How will we measure how much algae there is? This is the hard bit. I haven't got a machine I can just turn on and it tells me. So I need to use a piece of equipment. So yeah, do you know what piece of equipment we use? Because I've got an entire tree. I need to designate a small area. So we use something called a quadrat, a frame of known area, not a quadrant. Quad for rat, R-A-T. Quadrat, one word. Why do we need to measure percentage cover rather than count algae? Well, I can't see algae. I can see the carpet of algae, but I can't see individual algae, so I can't make them out. Therefore, I do percentage cover. So yeah, they're too small to see individual algae. How will we measure light? So this is where it's easy. We just use a light meter or a light sensor. Yeah, it's easy. There's a machine for it. Number 3, where on the tree should we measure the algae? For how long should we measure light? If I measure it for a second, it's changing throughout the day. Sun comes up and sun goes down. Changes from day to day with weather and cloud cover, doesn't it? And it changes from month to month with seasons. So it's crucial that I measure it, that I think about that when I'm actually measuring it. Where on the tree? Yeah, so 1 meter is a good suggestion. If we just said measure it on the tree, it's too vague. I've put 1 meter. It's a sensible suggestion. 5 meters wouldn't be sensible. It's too high. you couldn't get an accurate measure of the algae. Okay, and it'd be dangerous. Two, if I put it at the base of a tree, where you can see here there's like grass at the bottom of the tree, that might obscure the algae. So yeah, 1 metre is a sensible suggestion. How many trees should we measure? Which will give a more confident conclusion, 5 trees or 10. The more data you have, the more confident you become. So yeah, 10 trees is better than 5. Okay. In science, a lot of people think 3. Like you're doing chemistry and physics, you'll repeat something to get 3 sets of data so you can find the anomaly. Check for anomalies with 3. So 3 is the minimum number you can find an anomaly with. When we're dealing with living things, we often need a lot more data. So yeah, 10 trees, minimum. I'll be doing 20. Should we repeat the measurement on different days or just pick? Yeah. Or just pick a sunny day. Again, it doesn't matter as long as you are, taking it into account. Both might answer the question. What should we do with the data once collected? So you'll have lots of data of light intensity. You've measured that, and percentage cover. You've measured that. So these are 2 dependent variables. And if there's 2 dependent variables, we draw a scatter graph. Okay, so line graph is when you've got independent, dependent. Like I've controlled the temperature and I'm measuring how much gas is produced or something. That's your typical line graph with a line of best fit. With scatter graphs, they can go either way around. Axes don't matter. You plot the points and then you look for a correlation and show that with a line, a straight line. You don't have a curvy line. You do have a curvy line on a line of best fit for a line graph. And then how I record data that is free of bias. So free of bias, if there's 100 trees and I want to pick 10 of them, how do I pick 10? Free of bias. Do I close my eyes and wander around and the one I bump into I pick? No, we don't. We number the trees and we use a random number table to pick them. So we need to get randomness, but humans can't be random. I can't say, right, go and find a tree and be completely random about it. Yeah, we can't. Yeah, no, name a vegetable. Name a random vegetable. Carrot. Everyone says carrot. Yeah. Name an animal. Cow. Everyone says cow. Yeah. Humans find it very hard to be random. If you've not said carrot or cow, well done. There are the 2 that popped into my head. So anyway, number 2. So free of bias, we have to get randomness into it, and we have to use something called like a random number table. Next is accuracy. If accurate, the percentage cover will be close to the true value. So I need to measure the algae and it needs to equal what's there. Okay, I'm going to come to that in a second. On the next slide, we'll hit accuracy. Reliable is a pain because GCSE science, they say don't use the word reliable. We should be using the word repeatability and reproducibility. And those 2 words are brilliant for practicals that you do in a lab. Yeah, because if it's repeatable, every time I do it, I get the same result. Every time I roll the ball down the ramp, I get the same data. It's very repeatable. Okay, reproducible, or when that group over there roll a ball down a ramp, their data is comparable to mine. It's reproducible. Okay, so I can understand why we don't want the word reliable for most of science. However, the word reliable, kind of, repeatability and reproducibility don't really work with this because we're not just going to keep measuring the same part of a tree. We're going to measure many trees. This is where I talk about being confident. Yeah, that it's anything that sticks in your head that means that you need to measure lots of trees. 1 tree is not enough. It might not be a typical tree. 3 trees, again, I've got a bit more confidence. 10 trees, I'm more confident. 100 trees, I'd be very confident. Yeah, in my conclusion, because I'd have lots of data. So again, it's a bit of an area where we're kind of stuck in biology. That there's no really suitable word. I'd still use reliable in my answer, but I'd also talk about being confident, just to make sure. Anyway, here's the tree trunk zoomed in, and there's my quadrat, a frame of known area, and I've split it into 25 little squares. And this is where I can spend ages measuring the percentage cover, or I just have a simple rule that says if it's got less than half in, I don't count it. So those 5 squares I've deemed that they have less than half full of algae. I'm not going to count them. So each square is 4%, each small square, because I've got 25 squares, and 25 times 4 is 100. And therefore I've got 20 squares that have got covering them, so I times it by 4, 80%. That's my rule for doing percentage cover. And because it's a simple rule, we're likely to agree. So when I count it and my partner counts it, we're likely to get the same answer. And we count that as accurate if we agree. If I didn't have those subdivisions, we'd unlikely to say the same value. I always says 75%, he says 82%. Yeah. So by having lines, it means that we can be more accurate. Okay. I could be more accurate if I had a 10 by 10 grid. So each square would be 1%, but that would take longer, and time is money. Again, not for us when we're practicing these skills, but for a professional ecologist, it would mean, that they'd spend longer. Therefore, they might only be able to count 3 trees, whereas I can count 20 trees using my method in the same time, and therefore I've got more data. It might be less accurate tree to tree, but because I've done more trees, I think that's better. But the other person could argue that they're better, and that's how science works. We all make our case, and then we criticize each other. We don't take it personally because we all want to improve and get better. More things should be like science in the other world, I always think. Anyway, how would you show that dandelions grow more in soil with a low pH? And I want you to plan a method. So again, you can have a go at this. You can pause the video. You need a quadrat, and you need 2 tape measures. And to help out, you can see this green field here. So here's a field. There's thousands of dandelions and a variable pH of soil. And you're going to put 1 tape measure on one axis of the field and 1 tape measure on the other axis of the field. Try and have a think why you need them. This is a pH probe and meter. You stick this into the soil, it gives you the pH. Dead easy. If you know the name of the technique, because there's 2 techniques you need to know, brilliant, pop it down. If you don't, that's why we're doing this, to find something that you don't know. Everything must be specific. So it doesn't need to be a perfect method by any means, but how many areas in the field are you going to measure? Yeah, if you don't know where to start by putting that down. Because if you put that down, that's better than the person who will just say, sample many areas in the field, or even vaguer. You need to ensure accuracy for your bias and reliability or confidence, and answer the full question. So pause it now, because I'm going to go through onto the next slide and go through a perfect 6-mark answer. So the answer, again, the technique is called random sampling. Okay, it's often worth putting that first. Also, if it's a 2-mark question, then it's the answer as to how you do this. You can do random sampling with quadrats. You're going to place 2 measures at 90 degrees to each other on the edge of the field. I told you that. You're going to look up random coordinates from a random number table. So because I've got an X and a Y axis with tape measures, I can just look up a random coordinate. I have a little book full of random numbers, and I use them for coordinates. I placed my quadrat, I put the size of it. I didn't need to. The coordinates, and I'm going to count the number of dandelions in the quadrat. The number of people who miss out, count the number of dandelions in the quadrat. Okay, it's unbelievable. So well done if you put that. And double check for accuracy. That's all you need to do. Yeah, do it once, do it again to check that it's accurate. 5, place the pH probe at a depth of 10 centimeters. That's specific. That's good. In the center of the quadrat, perfect. And record the pH. So I know exactly where to put the probe, at what depth, perfect. And I repeat steps 3 to 5 in 19 other locations. You might have put 9 other locations to get 10, because people tend to default to 3 or 10. I always think get 20. No one's going to make you do it. 20 bits of data is more, gives you more confidence than 10. Then I'm going to plot the pH of each sample size against the number of dandelions at that location, and then I get a scatter graph. Done. That gets 6 marks. Everything's got like a number to it. It's therefore specific. Someone could follow this and do it, and they won't have to ask any questions. A perfect 6-mark answer. Right. Then simple math skills. You need to know basic geometry. So I want you to have a go at this. Again, you can pause it again. How many dandelions in the bizarrely shaped field? Give your answer to 2 significant figures. So I've got, there's like a eaten out rectangle with a triangle. So yeah, you need to know areas of circles and areas of triangles and areas of rectangles. They're skills you need to have. It says after random sampling in 100 locations, the student had a mean number of dandelions of 8.3 per 0.25 meter squared quadrat. That's a quadrat with that's half a meter by half a meter. If it's square, so again, have a go. How many dandelions would there be? Right, I'm going to go through the answer now. Hopefully you paused it, not just waiting to watch me do this. So yeah, the area of the rectangle, 150 times 50. That's the intact rectangle before I deduct the semicircle. That's 7,500 meters squared. Area of semicircle is half pi r squared. The number of people who forget to halve the diameter is quite high. People who forget to halve the answer to get a semicircle is quite high. I'm going to call that B. That's 982. And then the area of the triangle is, well, it's half base times height. So 30 times 50, 1,500 meters squared. So the total area is the rectangle A minus the semicircle B plus the triangle C, 8,018 meters squared. And my quadrat is a quarter of a meter square. So I divide them to find how many quadrats would fit into the entire area, and then times it by the mean number. So that's 8,018 divided by the area of the quadrat. Therefore, my units disappear. Times by 8.3, I get 266,198 dandelions, which to 2 significant figures that people forget to finish off the question is 270,000 dandelions. Okay, so if you got that, 5 marks at GCSE. Thank you for getting all that right, which is what our students are really good at. Is like, common sense, basic maths. Nothing's hard here. It kind of sums up maths and biology. Now, in physics, a physicist would say that the mean number is 8 because they've measured dandelions and you can't have point of a dandelion. Okay, so a physicist would say 8. In biology, we earn a decimal place when we have lots of data. Okay, it's like the average family size in the UK is 2.4 children. Okay, if I didn't have that 0.4, I couldn't keep track of the average family size, how it changed from like decade to decade. If it used to be 2.4 and now it's 2.3 and then it's moving to 2.2, it's important that I have that decimal place because I can keep track of it. A physicist would have it's 2, it's 2, it's 2, it's 2. They would just round down to 2 every time. Okay, so again, this is another area like reliability where we differ from physics and chemistry, where you can have an extra decimal place if you've got lots of data. And here I've got 100 sample sites, so I've earned a decimal place. Okay, again, there's not a hard and fast, clear-cut rule of when to do this. You do it when common sense shows that it's important, it's there. So if I didn't have that 0.3, it would really affect my total number of dandelions in the area. Okay, so yeah, there are rules in science, but some of them we have to kind of bend slightly for our different kind of disciplines. And biology is the science where there's more kind of debate, well, not debate, an answer's not kind of set. There's lots of factors. Biologists are very good at awkward data. In our practicals, there are lots of variables that some of them are controllable, and some of them aren't. In physics, they tend to want to control everything so that there's only one independent variable. Everything else is controlled. They're measuring their dependent variable. Again, it's very hard to kind of control, like mice running around a maze and things. I can get them the same age, but I can't get. They're going to have genetic differences. One might just have woken up in a mood. Again, it's tricky. Anyway, well done if you got 270,000 there. Right, next technique. It says, I think yellow flowering plants increase in numbers with altitude up this hill. That's my observation. And it does look like I can't see hardly any yellow flowers at the bottom, and there's a sea of yellow at the top. So I want to measure this. I want to measure a distribution, how something changes from the bottom to the top of the hill. So this isn't random sampling. I'm not going to sample at various places in this area. because that would just measure how many yellow flowers are in the area. I want to measure how it changes. Okay, so again, you can just kind of watch this because I'm just going to go through it. We do something called a transect, where we lay a tape measure along the bottom of the hill and we pick a random starting point. So my first tape measure runs along the bottom here. Then I use a random number table and I pick a random start point, or 3. So I'm going to start at meter 3 here. Then we run a tape measure from the starting point up the slope. So I then go up the slope with a tape measure. Okay, up is important. You have to give a direction when you do a transect. I'm going to place my quadrat at 0 meters along the transect line. So there's my imaginary transect line that's not shown, and I pop a quadrat at the bottom here. And I count the number of yellow flowers as we did before, double check for accuracy as we did before. Then we repeat steps 4 and 5 every 20 meters. So it's systematic. I don't do any more random placement of quadrats. I start at 0, go to 20, 40, 60, 80, 120. There's a systematic measuring up the hill. Okay, and then we do all that again at some other random points. So I end up with 3 transects, and I do a mean of parallel transects. Okay, so yeah, this is called a transect. You still use a quadrat. You still bring in randomness by having a random starting point. And it's free of bias because you're doing it at predetermined distances. But that's, the rest of the skill is the same. So why do we do 3 transects rather than 1? Well, 1 transect might go along a path or something. It might not be representative of the entire field. If I do 3, I get more confidence. Okay. Why do 3 rather than 10? Well, 10 transects would take ages, and yeah, so 10 would take too much time, and yeah, so there is a time thing. A professional ecologist is paid, and they'll be paid to do so many hours, and therefore they have to do some, get reliable data that's accurate, free of bias, in the time allowed. Okay, if you're going to build a house on some green belt land, an ecologist has to come along and survey the land to make sure there's no protected species there, and they write a report. If you want a job where you're out and about collecting data, wearing an anorak in the good old British weather, and then you go back to your office and you look at your data, manipulate it using, not as in change it, as in you use a computer to kind of make a graph of the data and calculate and do some statistical work, write a report, submit the report, then ecology might be for you. Again, there's lots of government jobs where you're an ecologist, where you're employed to make sure that the land near the factory isn't polluting the land, and therefore you can survey the land and show that the protection that you're putting in is having the desired effect. Again, there's loads of jobs in ecology. I don't know how well paid it is, but again, maybe you've never thought about it. Again, the thought of being inside all day would drive me mad. Oh, the thought of being outside all the time, oh, that'd be bad. Oh, a bit of both though. Not many jobs where you get to go in and out, and you get to be in your scruffs, but then you get to put a suit on when you're kind of presenting to the company or something. Who knows? Anyway, right, we're just going to pop through a couple of these questions. Again, some classes work faster and some classes work slower, but I just want to work through a couple of these just to show you how easy it is. So some students study bluebell plants growing in 2 different habitats. Habitat A was a sunny field next to woodland, and habitat B was shady and moist woodland. Right, a bluebell plant can have several flowers on 1 stalk. The student counted the number of flowers on each of 40 bluebell flower stalks growing in each habitat. The bar chart shows the result. So I've got a plant stalk that might have a few flowers, or it might have many flowers. So make sense of the data. Habitat A is sunny, habitat B is shady. So in the sunny field, this bar here that says 20, well, there's 2 stalks that have got 20 flowers on them. Okay, so that's a highly flowered stalk. There were two of them out of the 40 that I measured. 2 of them had lots of flowers. 1 of them only had 5 flowers. So I'm making sense of the data. Yeah. In the shady area, well, they've got less flowers in general. Well, there's one here that's only got 2 flowers on it. and none had above 10. Okay, so let's have a look at the questions. Says a student wanted to collect valid data. Describe how the student should have sampled the bluebell plants at each habitat to collect valid data. There's a big field. How am I going to? Which plants am I going to measure? Well, we use random sampling with quadrats. We use a quadrat to kind of pop it down to say these. I'm going to count them in this quadrat, and we make sure that we place that in a random way. Next question, the student used the bar charts to find the mode for the number of flowers per stalk in 2 habitats. What is the mode, the most common, for the number of flowers per stalk in habitat B? Well, I got this wrong the first time I put it. I put 12. I thought, 12 mode. I'm kind of used to seeing the mode on the y-axis, but that just means there's 12 plants had 6 flowers per stalk. Fortunately, I went back to check the accuracy of my PowerPoint, and therefore I spotted it, so I know 6 is correct. So 6 flowers was the mode. It was 11 for the other one though, so almost twice as much. The student suggested the following hypothesis. The difference in the modes between the 2 habitats is due to the plants receiving different amounts of sunlight. Suggest why. Well, why means I need to give a reason. Why do I get more flowers in a sunny environment? Well, more light equals faster photosynthesis. Yeah, it's a question about light, isn't it? If the question is about light, going back to the first slide we did, if the question talks about light, the answer is going to be photosynthesis. How can it not be in biology? I can't think of it. It's if the question is about, I guess it could be about sight, but we haven't done the eye yet. Something to look forward to for next year, the eye. But generally, if it's plants and light, it's photosynthesis, isn't it? Next, suggest how the student could test their hypothesis for the two habitats. They think that there's a difference in light. Well, I need to measure the light, don't I? In lots of different places at the same time of day or something like that. So they're going to use a light meter or measure light intensity in both habitats. Okay, so they're going to measure the light intensity in both habitats, and then it needs to be controlled, so you need to measure it at the same time of day. Remember, light can change hour by hour, day by day, month by month, second by second, can't it? So yeah, you need to do it at the same time. You'd probably set them both up, and then you'd leave them recording on like a data logger type thing. Then you'd come back the next day so you can compare exactly the same times of day, something along those lines. Next, suggest how receiving more sunlight could result in the plants producing more flowers per stalk. So now, I've already said photosynthesis, but how does more photosynthesis equal more flowers? Okay, what do we know? Well, more photosynthesis equals, what's the product of photosynthesis? What do they use? Yeah, well, it's glucose. There's more glucose, and therefore they can grow faster. Yeah, more glucose, 1 mark, the semicolon shows the end of the first marking point for growth. Again, I'll probably put they do more photosynthesis in case I got the previous one wrong. More photosynthesis, therefore more glucose. More glucose means they can join glucose molecules more together to form more cellulose or more starch or more protein. Could say anything, but they only want growth. Last one we'll zip through because it's because they like asking about tides quite often. So on a rocky shore, when a tide goes in and out, organisms are exposed to the air for different amounts of time. Yeah, so we've got tides because of the moon's gravitational pull on the Earth. The oceans kind of bulge. So twice a day we end up with tides, a high tide and a low tide. And if you're high up the beach, you're going to be covered in water at high tide, and then you're going to be exposed to the elements, to the dryness and sunlight and heat when the tide goes back out again. So on hot, windy days, when the tide is out, the concentration of salt solution in rock pools may become very high. Yeah, so the fresh seawater collects in rock pools, it goes out, it leaves a pool of seawater behind that's going to evaporate the water, and therefore it gets saltier and saltier, increased salinity. What term is used to describe organisms that can survive in severe conditions, such as very high salt concentration, salt solution? Well, that's called an extremophile. Oops. Oh yeah, extremophile. I thought I spelled it wrong. Yeah, that is correct. Extremophile. Hopefully you did that in your remote learning. Then periwinkles are types of snails. Students survey different types of periwinkle living on a rocky shore. The diagram shows results of the student survey. The highest position that the sea water reaches on the shore is called the high tide level. Oh yeah, it's there. Each bar represents the range of habitats of each type of periwinkle. Right, so I've got 4 types of them. And this one lives mainly at high tide. These 2 live at low tide. Good. Which 2 types of periwinkle are likely to compete with each other to the greatest extent? Explain your answer. Well, it's going to be those 2, isn't it? It's going to be common and the flat because they live in the same area. If they live in the same place, they're going to be competing with each other. Makes sense. Nice and easy. The small periwinkle can survive much nearer to the high tide level than the flat one. Okay, suggest 2 reasons why the flat periwinkle cannot survive near to the high tide level. This is where a physicist would go, oh, what's it? Oh, they would hate our market because there's just so many. Any sensible suggestion. That word suggest, and this is the last one we'll do. But the word suggest means anything sensible, and what a lot of students are in is, and I fall into it as well, is I'm trying to think of the correct answer. What is the correct answer? But I barely know about these animals. But it's anything sensible. It could be the wrong food. They're exposed to predators that they're not used to, so they might have poor camouflage for being out on the beach. They can't tolerate extended exposure to the air or reduce submersion in seawater, so they need to be covered in seawater for most of the time. They can't cope with temperature change or dehydration. They can't tolerate high salt concentration. They can't compete with the small periwinkles. They can compete with the common ones though, but these ones are like, oof, don't mess with these. They'll just eat all the food. Okay, so yeah, don't be scared by the words suggest. Okay, so hopefully that was useful. Again, nothing's technical. Extremophile's technical, but at the end of the day, you need to be able to write a random sampling with quadrats answer and a transect using quadrats answer. Random sampling is you measure what's there, whereas transects, you measure what's there, but you want to know how it's spread, so you move along a line. And if you just remember those 2 things, then again, there's not a lot to revise for this. It's just little tips means that you'll hopefully get more marks. Good.